Hello and welcome, dear students. Today we are going to discuss about the topic common mechanisms of epigenetics. The main objectives of today's lecture are to know about the basics of epigenetics, to know about the mechanisms of epigenetics, to discuss about the chromatin remodeling, to discuss about DNA methylation, and finally to discuss about the histone modifications. Dear students, let's start with the introduction first. The term epigenetics was first used to refer to the complex interactions between genome and the environment that are involved in development and differentiation in higher organisms like humans. Today, this term is used to refer to heritable alterations that are not due to change in the DNA sequence itself. Rather, epigenetic modifications or tags such as DNA methylation and histone modification alter DNA accessibility and chromatin structure, thereby regulating patterns of gene expression. These processes are crucial to normal development and differentiation of distinct cell lineages in the adult organism. They can be modified by exogenous influences and as such can contribute to or be the result of environmental alterations of phenotype or pathophenotype. Importantly, epigenetic programming has a crucial role in the regulation of pluripotency genes which become inactivated during differentiation. Epigenetic mechanisms contribute to the regulation of physiology and diseases by changing gene expression, and epigenetic changes occur as a result of the interplay between DNA and environmental factors. Unlike permanent change in DNA sequences that change amino acid sequences or gene expression, epigenetic regulation can respond dynamically to environmental and lifestyle factors. However, this regulation may have more stable, long-lasting effects than do transient change in gene expression or cellular regulation independent of change in gene expression. The fundamental importance of epigenetic mechanisms in biology is illustrated best by striking epigenetic phenomena such as the presence of 200 cell types in the human body that are distinct molecularly and phenotypically, although nearly all of them share identical DNA sequences. Another striking illustration of epigenetic mechanisms' biological importance is the inactivation of one of the X chromosomes in female cells. Increasingly strong evidence supports epigenetic mechanisms' significant role in the development of hypertension. Nevertheless, many fundamental questions remain to be addressed. Epigenetics is the study of heritable and stable changes in gene expression that occur through alterations in the chromosome rather than in the DNA sequence. Despite not directly altering the DNA sequence, epigenetic mechanisms can regulate gene expression through chemical modifications of DNA bases and change to the chromosomal superstructure in which DNA is packed. Briefly, negatively charged DNA is packaged around a positively charged histone protein octamer which contains two copies of histone proteins H2A, H2B, H3 and H4. The nucleoprotein complex is a nucleosome, the basic unit of chromatin. The nucleosomes of a continuous DNA polymer are connected by linker DNA and the complex is stabilized by histone protein H1. The aggregation of chromatin results in the formation of a chromosome. The chromatin of a chromosome exists as either loose, transcriptionally active eumachromatin or dense, transcriptionally inactive heterochromatin. Chemical alterations to histone proteins can induce the formation of either the open euchromatin state, which facilitates gene expression by allowing transcription factors and enzymes to interact with the DNA, or the closed heterochromatin state, which suppresses gene expression by preventing initiation of transcription machinery. In addition to histone chains, DNA methylation is an epigenetic mechanism associated with gene silencing when the methylation occurs in CpG islands or promoter sequences. Further, non-coding RNA sequences have shown to play a key role in the regulation of gene expression. These epigenetic modifications can be induced by several factors, including age, diet, smoking, stress, and disease state. Epigenetic modifications are reversible, but they rarely remain through generations in humans despite persisting through multiple cycles of cell replication. Dear students, let's now discuss about the mechanisms of epigenetics. Epigenetic mechanisms are sensitive to environmental effects and key participants in shaping an adult phenotype. Several types of epigenetic inheritance systems may play a role in what has become known as cell memory. However, not all of these are universally accepted to be examples of epigenetics. The genome within each cell is identical. However, the terminal phenotype is contributed by manifestations of epigenetic markers in the genome that lead to deviations in their gene expression profiles. These deviations are regulated mainly by DNA methylation as well as modifications of histone and RNA associated silencing. Dear students, let's now discuss about the covalent modifications. The covalent modifications of either DNA, for example, cytosine methylation and hydroxymethylation, or of histone proteins like lysine acetylation and arginine methylation, serine and threonine phosphorylation and lysine ubiquitination and summorylation play central roles in many types of epigenetic inheritance. Chromatin remodeling is not always inherited, and not all epigenetic inheritance involve chromatin remodeling. Because the phenotype of a cell or individual is affected by which of its genes are transcribed, heritable transcription states can give rise to epigenetic effects. There are several layers of regulation of gene expression. One way that genes are regulated is through the remodeling of chromatin. Dear students, let's discuss the chromatin remodeling. Chromatin remodeling is accomplished through two main mechanisms. The first way is post-translational modification of the amino acids that make up histone proteins. Histone proteins are made up of long chains of amino acids. If the amino acids that are in the chain are changed, the shape of the histone might be modified. DNA is not completely unwound during replication. It is possible then that the modified histones may be carried into each new copy of the DNA. Once there, these histones may act as templates, initiating the surrounding new histones to be shaped in the manner by altering the shape of the histones around them. These modified histones would ensure that a lineage-specific transcription program is maintained after cell division. The second way is the addition of methyl groups to the DNA, mostly at CpGA sites, to convert cytosine to 5-methyl cytosine. 5-methyl cytosine performs much like a regular cytosine pairing with a guanine in double-stranded DNA. However, when methylated cytosines are present in CpG sites in the promoter and enhancer regions of gene, the genes are often expressed. But when methylated cytosines are present in CpG sites in the gene body in the coding region, excluding the transcription start site, expression of the gene is often enhanced. Transcription of a gene usually depends on a transcription factor binding to a 10 base or less recognition sequence at the enhancer that interacts with the promoter region of that gene. there is frequently a reciprocal relationship between DNA methylation and histone lysine methylation. Mechanisms of heritability of histone state are not well understood. However, much is known about the mechanism of heritability of DNA methylation state during cell division and differentiation. Heritability of methylation state depends on certain enzymes that have a higher affinity for 5-methyl cytosine than for cytosine alone. Dear students, let's discuss about the DNA methylation process as an epigenetic mechanisms. DNA methylation frequently occurs in repeated sequences and helps to suppress the expression and mobility of transposable elements. Because 5-methyl cytosine can be spontaneously deaminated, replacing nitrogen by oxygen to thymidine. CpG sites are frequently mutated and become rare in the genome except at CpG islands where they remain unmethylated. Epigenetic changes of this type thus have the potential to direct increased frequencies of permanent genetic mutations. DNA methylation patterns are known to be established and modified in response to environmental factors by a complex interplay of at least 3 independent DNA methyltransferase enzymes, that is DNMT1, DNMT3A, and DNMT3B, the loss of any of which is lethal. DNMT1 is the most abundant methyltransferase enzyme in somatic cells, localizes to replication foci, has a 10 to 40-fold preference for hemimethylated DNA, and interacts with the proliferating cell nuclear antigen. By preferentially modifying hemimethylated DNA, DNMT1 transfers patterns of methylation to a newly synthesized strand after DNA replication, and therefore is often referred to as the maintenance methyltransferase. DNMT1 is essential for proper embryonic development, imprinting, and X inactivation. To emphasize the difference of this molecular mechanism of inheritance from the canonical Watson-Crick base pairing mechanism of transmission of genetic information, the term epigenetic templating was introduced. Furthermore, in addition to the maintenance and transmission of methylated DNA states, the same principle could work in the maintenance and transmission of histone modification and even cytoplasmic, that is structural heritable states. Dear students, let's discuss about histone modifications. Histone modification is the process of modification of histone proteins by enzymes, including post-translational modification such as methylation, acetylation, phosphorylation, and ubiquitination. These modifications constitute a rich histone code. Histone modification play a vital role in gene expression by modulating the degree of tightness or compaction of chromatin. Histones H3 and H4 can also be manipulated through demethylation using histone lysine demethylase. This recently identified enzyme has a catalytic active site called the Jumonji domain, or JMGC. The demethylation occurs when JMGC utilizes multiple cofactors to hydroxylate the methyl group, thereby removing it. JMGC is capable of demethylating mono, di, and trimethylated substrates. Chromosomal regions can adopt stable and heritable alternative states, resulting in bistable gene expression without change to the DNA sequence. Epigenetic control is often associated with alternative covalent modifications of histones. The stability and heritability of states of large chromosomal regions are suggested to involve positive feedback where modified nucleosomes recruit enzymes that similarly modify nearby nucleosomes. It has been suggested that chromatin-based transcriptional regulation could be mediated by the effect of small RNAs. Small interfering RNAs can modulate transcriptional gene expression via epigenetic modulation of targeted promoters. Dear students, let's now discuss about the RNA transcripts. Sometimes a gene, after being turned on, transcribes a product that directly or indirectly maintains the activity of that gene. For example, HNF4 and MYOD enhance the transcription of many liver-specific and muscle-specific genes, respectively, including their own, through the transcription factor activity of the proteins they encode. RNA signaling includes differential recruitment of a hierarchy of generic chromatin modifying complexes and DNA methyltransferases to specific loci by RNAs during differentiation and development. Other epigenetic changes are mediated by the production of different splice forms of RNA or by formation of double-stranded RNA. Descendants of the cell in which the gene was turned on will inherit this activity even if the original stimulus for gene activation is no longer present. These genes are often turned on or off by signal transduction, although in some systems where sync or gap junctions are important, RNA may spread directly to other cells or nuclei by diffusion. A large amount of RNA and protein is contributed to the zygote by the mother during oogenesis or via nurse cells, resulting in maternal effect phenotypes. A smaller quantity of sperm RNA is transmitted from the father, but there is recent evidence that this epigenetic information can lead to visible changes in several generations of offsprings. Dear students, let's now discuss about the non-coding RNA. Non-coding RNAs that are not translated into proteins can be divided into housekeeping non-coding RNAs and regulatory non-coding RNAs. RNAs that has a regulatory role in mainly divided into two categories based on size: short-chain non-coding RNAs, including siRNAs, miRNAs, and piRNAs, and long non-coding RNAs, that is LNC RNAs. In recent years, a large number of studies have shown that non-coding RNAs play a significant role in epigenetic modification and can regulate expression at the level of the gene and the level of chromosome to control cell differentiation. With the attention on non-coding RNAs in the etiology of disease, the non-coding RNA has become a hot issue in modern genetics research, especially as a new mechanism of epigenetic regulation. A non-coding RNA is a functional RNA molecule that is transcribed from DNA but not translated into proteins. Epigenetic related non-coding RNAs include miRNA, siRNA, piRNA, and long non-coding RNA. In general, non-coding RNAs function to regulate gene expression at the transcriptional and post-transcriptional level. Those non-coding RNAs that appear to be involved in epigenetic processes can be divided into two main groups: the short non-coding RNAs and the long non-coding RNAs. The three major classes of short non-coding RNAs as discussed are microRNAs, short interfering RNAs, and PV interacting RNAs. Both major groups are shown to play a role in heterochromatin formation, histone modification, DNA methylation, targeting, and gene silencing. Dear students, let's discuss the long non-coding RNAs. Many long non-coding RNAs can complex with chromatin modifying proteins and recruit their catalytic activity to specific sites in the genome, thereby modifying chromatin states and influencing gene expression. The majority of non-coding RNA transcripts belong to the group long non-coding RNAs. So long non-coding RNAs function in chromatin remodeling, transcriptional regulation, post-transcriptional regulation, and as precursors for siRNA. One particular subgroup of long non-coding RNAs are the large intergenic non-coding RNAs, or that is LINC RNAs, has been associated with chromatin modifying complexes which can target specific genome loci to promote specific epigenetic states. One widely known example of this is the role of X-inactive specific transcript gene XIST in X chromosome inactivation. This process involves two long non-coding RNAs, XIST and its antisense transcript TSIX, a negative regulator of XIST. Prior to differentiation, XIST and TSIX are actively transcribed due to H3K4 dimethylation of the XIST gene. In this state, XCL is a random event. Upon differentiation, XIST expression is elevated, resulting in XIST RNA coating the future inactive X chromosome, which triggers extensive histone methylation and chromosome inactivation. Dear students, let's now discuss about the microRNAs. MicroRNAs or miRNAs are members of non-coding RNAs that range in size from 17 to 25 nucleotides. miRNAs regulate a large variety of biological functions in plants and animals. So far, about 2,000 miRNAs have been discovered in humans, and these can be found online in a miRNA database. Each miRNA expressed in a cell may target about 100 to 200 messenger RNAs that it downregulates. Most of the downregulation of miRNA occurs by causing the decay of targeted miRNA, while some downregulation occurs at the level of translation into protein. It appears that about 60% of human protein coding genes are regulated by miRNA. Many miRNAs are epigenetically regulated. About 50% of miRNA genes are associated with CpG islands that may be repressed by epigenetic methylation. Transcription from methylated CpG islands is strongly and hereditably repressed. Other miRNAs are epigenetically regulated by either histone modifications or by combined DNA methylation and histone modification. miRNA has been demonstrated that the methylation of mRNA plays a critical role in human energy homeostasis. The obesity-associated FTO gene is shown to be able to demethylate N6-methyladenosine in RNA. sRNAs are small, highly structured, non-coding RNA fragments found in bacteria that control gene expression, including virulence genes in pathogens, and are viewed as new targets in the fight against drug-resistant bacteria. They play an important role in many biological processes binding to mRNA and protein targets in prokaryotes. Their phylogenetic analysis, for example, through sRNA, mRNA target interaction or protein binding properties, are used to build comprehensive databases. sRNA gene maps based on their targets in microbial genomes are also constructed. Dear students, let us discuss about the prions. Prions are infectious forms of proteins. In general, proteins fold into the discrete units that perform distinct cellular functions, but some proteins are also capable of forming an infectious conformational state known as prion. Although often viewed in the context of infectious diseases, prions are more loosely defined by their ability to catalytically convert other native state versions of the same protein into an infectious conformational state. It is in this later sense that they can be viewed as epigenetic agents capable of inducing a phenotypic change without a modification of the genome. Fungal prions are considered by some to be epigenetic because the infectious phenotype caused by the prion can be inherited without modification of the genome. The two fungal prions PSI plus and URA3 discovered in yeast in 1965 and in 1971 are the two best studied of this type of prion. Prions can have a phenotypic effect through the sequestration of protein in aggregates, thereby reducing that protein's activity. In PSI plus cells, the loss of the SUP35 protein, which is involved in the termination of translation, causes ribosomes to have a higher rate of read-through of stop codons, an effect that results in suppression of nonsense mutation in other genes. The ability of the SUP35 to form prions may be a conserved trait. it could confer an adaptive advantage by giving cells the ability to switch into a PSI plus state and express dominant genetic features normally terminated by stop codon mutations. Dear students, let's now discuss the nucleosome positioning. Eukaryotic genomes have numerous nucleosomes. Nucleosomes position is not random and determine the accessibility of DNA to regulatory proteins. Promoters active in different tissues have been shown to have different nucleosome positioning features. This determines differences in gene expression and cell differentiation. It has been shown that at least some nucleosomes are retained in sperm cells where most of but not all histones are replaced by protamines. Thus, nucleosome positioning is to some degree inheritable. Recent studies have uncovered connections between nucleosome positioning and other epigenetic factors such as DNA methylation and hydroxymethylation. Dear students, let's now discuss about the histone variants. Different histone variants are incorporated into specific regions of the genome non-randomly. Their differential biochemical characteristics can affect genome functions via their roles in gene regulation and maintenance of chromosome structures. Dear students, let's discuss about the genomic architecture. The three-dimensional configuration of the genome, that is the 3D genome, is complex, dynamic, and crucial for regulating genomic function and nuclear processes such as DNA replication, transcription, and DNA damage repair. Dear students, let's now understand the various causes that lead to epigenetic change. There are various causes that lead to the epigenetic change. The first one is development. Epigenetic variations cause the cells of an embryo to develop and differentiate into cells having different specific functions. Second is the age. Throughout the natural course of a person's life, epigenetic changes occur. Third is the lifestyle. Change in a person's behavior or surrounding environment can result in epigenetic change. Fourth one is the disease. Infections can cause epigenetic variations in your body. Certain epigenetic factors can increase susceptibility to disease like cancer. Cancer can also cause epigenetic abnormalities. Dear students, let's now conclude the lecture. Epigenetics is the study of mechanisms that are behind changes in gene expression that are not caused by changes in the DNA. These changes are called epigenetic variation or epigenetic changes. These epigenetic variations are either hereditary or a result of the environment within and surrounding an organism. This is why identical DNA or identical genes don't always result in identical outcomes. All the cells in our body typically have the same DNA sequences, so there must be something else that decides what phenotype is expressed in each part of our body. Epigenetic variations can turn genes on or off and therefore affect the protein production in the cells. This makes sure that different cells in our body produce only the proteins that are in accordance with their corresponding functions. Even though bone cells and muscle cells carry the same DNA sequences, proteins for muscle growth are not produced in bone cells. Beyond localized production of proteins suitable for specific cell functions, epigenetic variations are important because it allows our fixed DNA to adapt dynamically to different environments that we find ourselves in. There are different mechanisms that drive epigenetic variations and control gene expression. Three of the most important of them are DNA methylation, histone modification, and non-coding RNA. The study of epigenetics has the potential to give us valuable insights into how the genetic mechanisms of our body operate. From this, there can be a better diagnosis and treatment of diseases. Beyond human health, epigenetic research now also finds applications in agriculture and other fields. The advancements in technology in recent times make it a prime time for exploring the epigenome, and there is a long way to go before the opportunity is fully realized and utilized. Dear students, it was all about today's lecture regarding common mechanisms of epigenetics. Hope you have understood it well. See you next time with a new topic. Till then, take care. Goodbye.